All right, engineers, in this video, we're going to specifically focus on the mobilization of fats. Now, generally, where are these fats, and what do we categorize as fats? We technically categorize them as triglycerides. Okay, so now these fats are going to be stored within tissue cells in our body, and this is going to be one of the main storages of this actual fat. This is actually going to be the adipose tissue, but we're just zooming in on one cell, or you can also refer to this as an adipocyte, which is just a fat cell. So this right here is a adipocyte. So this is an adipocyte. An adipocyte is a fat cell, right? What we're going to do is we're going to look at how we're actually going to break this triglyceride down into its constituent units. What are the constituent units of a triglyceride? One is glycerol, and the other one is 3 fatty acid chains. So that's the goal of this video, is to go over exactly what mechanisms are triggering the breakdown of this triglyceride and how is that happening. Okay, so the first question to ask is, when is this happening? Okay, so this usually is happening in our body whenever we're in the fasting state. Okay, so this is usually occurring when we're in the fasting state. What is the fasting state? The fasting state is the state in which we haven't eaten in a while, right? So we haven't either, we're starving ourselves, or we're actually not taking in food. We're in the post-absorptive state. So what does that mean for our blood glucose levels? What does that mean? If you haven't eaten for a while, then you're not going to really have high blood glucose levels. So your blood glucose levels, as a result, are going to be low. Is going to be low. When blood glucose levels are low, what hormones or molecules are going to be released to be able to bring those blood glucose levels back up? There is many molecules, but we're going to focus on just a select few. Okay. The main one that you guys need to remember is within the pancreas. In the pancreas, there's actually these things called islets of Langerhans. They're clusters of endocrine tissue. And there's 2 different types of significant cells in this area, alpha and beta cells. The beta cells are releasing insulin, but that's when the blood glucose levels are high. Glucagon is releasing, alpha cells are releasing glucagon whenever the blood glucose levels are low. Easy way to remember that is, glucagon is being released whenever the glucose is gone or low. Okay, so now, the pancreatic alpha cells will release a hormone referred to as glucagon. Okay, so one hormone that we're going to release, this is going to be a very significant one, is glucagon. And this is going to be triggered based upon low blood glucose levels. Another hormone that can get released, whenever we're in stressful situations, our body is trying to be able to accomplish this way of being able to deal with certain situations in which our blood glucose levels are low. Maybe we have to be able to increase them in some type of fight or flight situation or stressful situation. Sometimes our body can activate the sympathetic nervous system. And the sympathetic nervous system has these preganglionic motor neurons within the lateral gray horn of the spinal cord from about T1 to L2. And those fibers can come out here into the adrenal medulla. And the adrenal medulla has specific types of postganglionic motor neurons of the sympathetic nervous system. They're consisting of things called chromaffin cells. And what they do is they secrete a chemical into the blood who's going to help us with this actual blood glucose levels. This molecule is going to be epinephrine. So epinephrine is going to be released, and he's released in very significant amounts from the adrenal medulla. About 80% of it is epinephrine. Very tiny amounts of it, about 20% of it, is going to be in the form of norepinephrine. So norepinephrine. Okay, so these are the main chemicals, but there is other molecules being released, like cortisol, like thyroid hormone, many other different types of hormones, growth hormone. We're going to focus on the 3 significant ones, the more important ones. Okay, so glucagon, epinephrine, and norepinephrine. What are they going to do? They're going to come to this adipocyte, this adipose tissue cell. They have receptors on the extracellular membrane, a G protein coupled receptor. What they can do is, let's say that I show here in green, this is representing these hormones. Okay, so this is, I'll put H for these hormones. And what could these hormones be? It could be norepinephrine, could be epinephrine, or could be glucagon. When these hormones are binding onto this G protein coupled receptor, that when it activates it, activates a G stimulatory protein. And then G stimulatory proteins bind onto GTP, which make them active. When they become active, they go and activate a specific type of effector enzyme that's present or embedded within inside of the cell membrane. This effector enzyme is called adenylate cyclase. And adenylate cyclase does what? It takes ATP and converts it into cyclic AMP. And then cyclic AMP goes and activates another enzyme. And this enzyme is called protein kinase A. Okay, why is all of this important? Okay, you see this protein kinase A? You know he loves to phosphorylate specific types of proteins within the body. So in this cell, there's special proteins. You know this, there's kind of like a little surrounding structure around this triglyceride, because you know there's many, many triglycerides within this adipose site, tons and tons and tons. This protein kinase A, you know there's molecules kind of surrounding this triglyceride. You see that maroon colored molecule? That molecule right there is called perilipin. So again, what is this molecule right here called? It's called perilipin. Perilipin is normally bound to another molecule right adjacent to it. You see this green molecule right there? That green molecule is called a CGI molecule. CGI molecule. So these two are literally connected to one another. Okay? So you have perilipin with CGI bound together. But what happens is this protein kinase A comes over here and puts phosphate groups onto this perilipin. So he phosphorylates this perilipin. When he phosphorylates this perilipin, the CGI, so look, we're going to put phosphates on this perilipin. When we put phosphates on this perilipin, the CGI disassociates. When the CGI disassociates, where does he go? So look, here is our CGI. He's going to go somewhere else. Where is he going to go? You see this enzyme right here, this brown scissor enzyme? That brown scissor enzyme is really important. And the reason why that brown scissor enzyme is important is because he's going to make the first cut into this triglyceride. And who's going to help him? You see this green molecule here? This green molecule here, this CGI, is going to come over here and it's going to bind with this enzyme and stimulate this enzyme. This enzyme is going to make the first cut on the triglyceride. What he's going to do is he's going to cut, you know the bonds here are specifically ester bonds. You have esters here because you have the carboxyl end of the fatty acid and then you have the alcohol, you have an alcohol in from the glycerol. What's going to happen here is that you're going to specifically cut that ester bond. When you cut that ester bond, guess what I'm going to release from this first point here? What am I going to release out of this first step? This first step is specifically going to release out a fatty acid. So from this reaction, the first thing that I'm going to do is I'm going to release out a fatty acid. So let's show that one fatty acid. Let's put one here to simplify and to signify that this is the first fatty acid. So now coming out of that is going to be specifically the first fatty acid molecule. We're just now going to put F-free fatty acid, and let's denote this one 1. Okay? So this is the first free fatty acid that we released, and this was from that point right there. So we're going to call this one, let's put a 1 right here on this one. Okay, what is this enzyme? This enzyme here is called Adipose triglyceride lipase. So again, what is this enzyme here called? ATGL is the abbreviation, and this enzyme is stimulating this step again, right? What is he doing? He's breaking specifically this ester bond between this free fatty acid and that glycerol molecule. When he breaks that bond, he releases out a free fatty acid. And this enzyme is called adipose triglyceride lipase. And he is activated whenever CGI, who is this molecule right here again? CGI, disassociates from the perilipin and activates this enzyme. Then, you know, protein kinase A, he's not picky. He's not going to be like, oh, I'm going to phosphorylate this guy, but not any of these other guys. He comes over here and phosphorylates these enzymes too. You know, these enzymes are also important too. So look, let's show this phosphates right here. There's phosphates added onto this enzyme. And this enzyme is going to make the next cut. But again, what's going to be released? Let's actually show what's going to be released as a process from this. So we cut away that fatty acid. As a result, let's show the next step of this reaction, what we're left with. Because this is what we refer to as a TAG, a triacyl glycerol, or a triglyceride. It's synonymous, okay? But from the breakdown, what am I going to have left over, guys? Well, I should still have the glycerol. So I'm still going to have the glycerol here. But all I'm going to have left now is going to be 2 fatty acid chains because I released a free fatty acid in that first step. Then what's going to happen is this enzyme is going to cut this next one. So look what this enzyme is going to do. He's going to come over here, and he's going to cut that part. He's going to cut this bond. When he cuts this bond, he's going to release another fatty acid. So look, I'm going to release, out of this step, I'm going to release another, and the second one, I'm going to release another free fatty acid. What is this enzyme called that's doing this? This enzyme is called, and this is again free fatty acid, we're going to denote it 2. This enzyme right here is called hormone-sensitive lipase. Okay, which you're going to denote HSL. So hormone-sensitive lipase is doing what? It's cutting this molecule here. What do you call this molecule when you have a glycerol and 2 acyl groups or fatty acyl groups? You call this diacylglycerol, or just DAG. So this is a diacylglycerol, or we call it a DAG. Who's breaking down this DAG and releasing a free fatty acid? Hormone-sensitive lipase, when he's phosphorylated by protein kinase A. Then, what are we going to have left over? So again, this was the first step. This was the first step, which was acted on by adipose triglyceride lipase. And then he released a free fatty acid. In the second step, going to this next enzyme, because we have one more enzyme left, in the second step, what did we do? We released another free fatty acid, which was accomplished by hormone-sensitive lipase. We have one last enzyme left. What do you think he's going to do, guys? What do we have left? We have this glycerol and one fatty acid. So now what is this guy going to do? He's going to see this last point here. And he's like, that ester bond look good. I'm going to come in there and break that ester bond. So he comes in and breaks that ester bond. When he breaks that ester bond, he's going to release 2 molecules out of this. So what are the molecules that are going to be released from the product of this reaction? So look at this. This would be the third step. From the third step, this enzyme, and we'll mention him in just a second, is going to release what? It's going to release the third free fatty acid. This was the third one. And what else is going to come out as a product? Glycerol. And you're also going to release out of this glycerol. This is accomplished in that third step. So now, what is this enzyme who's doing this? It's not even that bad. It's called monoglyceride lipase. Not that bad, right? Because this is a monoglyceride. This is a diacylglycerol. This is a triacylglycerol. Or a triglyceride, diglyceride, monoglyceride. It's that simple. Or we can call it monoacylglycerol. So this is a monoacylglycerol lipase, or a monoglyceride lipase. They're synonymous. Okay, let's recap on what happened in this. Again, when is the mobilization of fats going to be occurring? It's going to be occurring in the fasting state. And the fasting state is when we haven't eaten in a while, right? So we're not taking in food, we're not ingesting food, so our blood glucose levels are going to be low. Our body needs to have glucose supplying our muscles, our brain, and vital tissues in order for them to operate and function and produce ATP. In order for that to happen, our body has these islets of Langerhans, with specifically the pancreatic alpha cells that are releasing glucagon. And sometimes in stressful situations, whether more specifically acute stress, our sympathetic nervous system can get activated. And what will it do? It'll activate the adrenal medulla to release 80% of epinephrine and 20% norepinephrine. And these molecules will come to the area. And like I said, in very, very chronic stress or in certain situations like severe hypoglycemia, the zona fasciculata can also release cortisol. And our anterior pituitary can release growth hormone, and our thyroid gland is also able to produce thyroid hormone that can also facilitate these functions too. But like I said, we're only focusing on these 3. But do recognize that there is other hormones. What are these hormones doing? They're binding onto a G protein coupled receptor of the adipose tissue. When they do that, they activate eventually throughout all this second messenger system, protein kinase A. Protein kinase A phosphorylates what molecule? Perilipin. Perilipins are kind of surrounding this whole triglyceride, right? But they're bound to a CGI protein. Whenever this perilipin is phosphorylated by protein kinase A, the CGI molecule dissociates. When he dissociates, he comes over here and binds onto adipose triglyceride lipase. When this adipose triglyceride lipase is activated, he'll make the first cut on this triglyceride, or triacylglycerol. When he does that, he'll break this ester bond and release a free fatty acid. That's the first one. Then, what are we left with after this triacylglycerol? We're left with a diacylglycerol, 2 fatty acids in this glycerol. There's also going to be phosphorylation of this enzyme, the hormone-sensitive lipase. The hormone-sensitive lipase will then cut the diacylglycerol and release a second free fatty acid. What's left over? I should denote what this one is called. This one is called a monoacylglycerol, okay? Or monoglyceride. And what happens here? This monoglyceride lipase, we should show that he's also getting phosphorylated. So you know he's also getting phosphorylated. And when he gets phosphorylated, he becomes stimulated. And when he is therefore stimulated, he breaks the monoacylglycerol at the ester bond and releases two things: the third free fatty acid and a glycerol. And that glycerol and free fatty acids are going to go where? That's what we're going to lead into next, but we're not going to finish here. We're going to do that in another video where we talk about how they're being oxidized. But specifically, those free fatty acids, they're going to get pushed out here. So let's say I draw here free fatty acids, and this is representing how many of them? So far, 3. And then what else will we have? We'll also have glycerol. These molecules are going to get pushed in the blood. Now, glycerol is slightly soluble, right? He's a kind of a water-soluble molecule because he has some nice alcohol groups on him. Whereas free fatty acids are not very soluble at all. So we can't just transport these in the blood on their own. So glycerol can be transported in the blood kind of on his own. A little bit of it might be bound to transport proteins, but not very much. This free fatty acids, though, they have to be bound to a transport protein. So sitting over here, waiting for those free fatty acids is a special molecule. This molecule is waiting and he's circulating throughout our blood plasma. He accounts for about 60% of our plasma proteins inside of the blood. This molecule is called albumin. And albumin will bind these free fatty acids. And then what will he do with them? He'll take these free fatty acids to different tissues in the body. Like where? He could take these free fatty acids to the muscles. He could take it to the heart muscle. So this could be skeletal muscles, could be heart muscle, or he could even take it into the liver or other tissues that are going to oxidize it for what? You making ATP. And we'll talk about all of this process. Now the next question is, what about that glycerol? We didn't talk about that glycerol. This glycerol is going to be special because the glycerol can actually be taken up by muscle cells too, or different types of tissues and utilized for energy. But there's also something special about this one. I'm going to show it in a different color. It can actually be taken up by the liver. And through a process called gluconeogenesis, I can convert that glycerol into glucose. And again, what is this process here called? Gluconeogenesis. Okay? And so again, this could be glycerol. So glycerol can be utilized for energy because you can get funneled into dihydroxyacetone phosphate, and we'll see that in a second. But specifically, one of the special things about this, if our blood glucose levels are low, we're going to want to be able to raise those blood glucose levels. So how does our body do that? It can take this glycerol and do what? Take it into the liver or even your kidney's proximal convoluted tubular cells and convert that into glucose. If we make more glucose, what was the original stimulus? What was the problem? Our blood glucose levels were low. If we could take this glucose and contribute it into the bloodstream, what will happen to our glucose levels? They'll eventually come back up. So that's one way. But also, another thing is, the whole reason this is happening is because our body needs energy. When you're in the fasting state, you don't have a lot of glucose. So you don't have a lot of fuel sources. And so what your body's going to do is whenever your blood glucose levels are low, maybe you're not actually taking in carbohydrates, maybe you're on a ketogenic diet, whatever it might be, your body needs to find energy wherever it possibly can. And so what it's going to do whenever your blood glucose levels are low, if it doesn't have that glucose as a source of fuel, it's going to start breaking down fat tissue to utilize that for energy. Because fat is actually going to produce more energy than carbohydrates. You know, carbohydrates only produce about 4 kilocalories per gram, whereas fats is almost 9 kilocalories per gram. So they're very, very concentrated sources of energy, and we'll see that why. We'll see why. But the whole point here was the blood glucose levels were low. We can do two things with breaking down these fatty acids and this glycerol. One thing is that if you don't have those carbohydrates in your diet, whether because you're fasting or you have diabetes or you're on a ketogenic diet, whatever it is, your body's going to use these free fatty acids to produce ATP because your body needs energy to function. The second thing is that we can take a small amount of this glycerol into the liver or the kidney's proximal convoluted tubular cells and make glucose via gluconeogenesis to increase the blood glucose levels. All right, engineers, that pretty much covers everything in this video for the mobilization of fats. In the next video, we're gonna see exactly how these free fatty acids are getting broken down to produce ATP. All right, engineers, I hope you guys enjoyed this video. Hope you liked it. If you did, hit that like button, subscribe, put a comment down in that comment section. All right, engineers, until next time.